Let's move on to phagocytosis, cell eating. There are several different types of phagocytic cells in your body. We have macrophages, and depending upon whether the macrophage resides within an organ or moves around, it might have a different name. Macrophages located within the central nervous system are called microglial cells. Those that reside within the air sacs or the alveoli of the lungs are called alveolar macrophages. The microglial cells and the alveolar macrophages are going to remain in their organs. Dendritic cells are associated with your skin. Once they come in contact with a pathogen and eat it, they will then move to the lymph node to present antigens to T cells. We'll talk about those later. Stellate macrophages are macrophages that reside within the liver. In addition, the second type of phagocytic cell are your neutrophils. Recall, these are granular sites and are usually the first to arrive at a site of infection. How do phagocytes recognize harmful substances? You want to make sure that they are not eating normal, healthy cells that belong to you. It's important that they recognize which cells must be eaten so that they don't make a mistake. And they will use protein receptors on their membranes to do this. These protein receptors are called pattern recognition receptors, or PRRs. They are a type of receptor called toll-like receptors. There are several steps in phagocytosis. I encourage you to learn the steps first in order and then learn what occurs within each step. The first step is adherence. The phagocytic cell has to come in contact with molecules located on the cell that is to be eaten and bind with those molecules. Recall that the PRRs, the pattern recognition receptors, are located on the plasma membranes of the phagocytic cells. Those proteins must come in contact and bind to molecules that are located on the membranes of pathogens. These are called pathogen-associated molecular patterns, or PAMPs. These are molecules that your cells do not have. This allows the phagocytic cell to recognize this cell as being foreign. In this illustration, this is the pathogen, and that is the PAMP. The phagocytic cell is here with the PRR. When the phagocytic cell comes across a PAMP, it will bind to it, and that is adherence. That's the very first step in phagocytosis. In order for phagocytosis to occur, this first step must happen. In addition, phagocytic cells not only eat pathogens, but they will also, or foreign cells, they will also eat damaged or dead cells that belong to you, as well as abnormal cells, such as tumor cells. And those cells are going to express what are called damage-associated molecular patterns. So they'll have these molecules on their membranes. The PRRs will bind to the DAMPs as well. This figure illustrates a pathogen with the PAMPs. Those are the red balls. This is the phagocytic cell, and these are the PRRs. So the pattern recognition receptors come in contact with the PAMPs and they bind. That is the adherence, the very first step in phagocytosis. If I ask you what the purpose is of this step, it is to prevent a phagocytic cell from eating a normal, healthy cell that belongs to you. The second step is ingestion, and that will be bringing in the cell that is to be eaten. This is the phagocytic cell. These are the PRRs. Here is a bacterium, and on that bacterium you will have the PAMPs. Adherence has occurred where the PRRs on the phagocytic cell bind to the PAMPs on the bacterium. That initiates the pseudopodia, which is just where the cytoplasm begins to move forward, creating these extensions that will eventually surround the bacterium or whatever the cell is that's being eaten, and these two ends then will fuse with one another, bringing this into the cell in a compartment called a phagosome. So what this phagosome is, it is a vesicle that contains a phospholipid bilayer right here. And that phospholipid bilayer was actually the plasma membrane, because this thing is going to, these two will fuse together and this will create a phagosome. So with ingestion, the pathogen or the cell that is to be eaten will be brought in and be located within a phagosome. The third step is killing. That phagosome is going to fuse with a lysosome, and you learned about lysosomes in AP1. Recall that these are organelles that contain digestive enzymes, which are hydrolytic enzymes. Remember, hydrolysis breaks larger molecules into smaller ones. And when the lysosome and the phagosome fuse together, that creates a phagolysosomes. And now the hydrolytic enzymes break down the cell that was eaten into molecules. That is your killing. And once the killing has occurred, you now have to eliminate all of the molecules that came about from the killing by exocytosis. So you've broken down the cell into molecules, and now you get rid of the molecules by exocytosis. In the adaptive immune system, T cells or T lymphocytes need to have their antigens presented to them in order for them to recognize and bind with them. A macrophage can take some of these molecules and they can function as antigens and bind them to proteins on their plasma membranes for presentation. We'll talk about that a little bit later, but this is for T cell activation. This is a good illustration, just going over phagocytosis again. Here's my pathogen. See if you can remember what these molecules located on the membranes of the pathogens are. These are your PAMPs. If this was a damaged cell that belongs to you or a cancer cell, they would be DAMPs, damage-associated molecular patterns. Can you name these located on the phagocyte? These are the PRRs, or the pattern recognition receptors. So this first step is adherence, where the PRRs bind with the PAMPs, and that stimulates the formation of the pseudopodia and phagocytosis. The pseudopodia will continue to grow, and remember, this is all plasma membrane, which is a phospholipid bilayer. And then these two ends, as the pseudopodia continue to grow, will fuse together, and you'll have ingestion, and now the pathogen is located within a phagosome. You're going to merge that phagosome with lysosomes, which have the digestive enzymes, and when you do that, you form a phagolysosome. The digestive enzymes will break down the cell that was ingested, and now you have to eliminate by exocytosis all of the molecules that are the remains of the cell that was digested. Recall that in the adaptive immune response, macrophages can take some of these molecules, which will function as antigens. Antigens are usually proteins, glycoproteins, that can be either self-antigens which will identify cells as belonging to you, or foreign antigens. And in this case, because this is a bacterium, these would be foreign antigens, these molecules here, and they would be placed on proteins on the outer surface of the macrophage. And we'll talk about that a little bit later. Dendritic cells will be doing this as well. This concludes this module. As with the first module, I'm going to encourage you to learn this material before you move on.